thank you for helping me out so much. So part of our partnership in doing the research workshop and the family sessions together, a lot of conversation. Are families going to understand the content in the research workshop? No, probably not. But Dr. Yin and Dr. Balistreri has graciously agreed to summarize the research workshop into layman's terms and present it back to us as families. So they're going to share the summary of the research workshop now. Thank you.
Sort of a yin and yang, I guess, right? So thank you all. It's a pleasure to not only share these thoughts with you, but also to share my perspective. I've been doing this for 50 years, and I have to tell you, like Rodrigo said, I'm leaving here very charged, because I'm going to start with the bottom line. What we heard in our research workshop, which we'll describe in a minute, is a high volume of very focused research on this disease, PFIC. This is what we've hoped for, and thanks to all of you, we are continuing to move ahead. We've heard about different approaches to the way to study these disorders, and the answers will clearly emerge. I was always taught that if you're not getting answers, you're not asking the right questions. And I think we clearly are now asking the right questions with the right models. And the word that I would end up with is hope. Like I say, I have 50 years of doing this, but I can't recall a time when there's been such many major advances in such a short period of time. When we started our transplant program in 1985, the headline in the paper, they interviewed us and they said, Cincinnati Children starts a liver transplant program. And I said, I'm looking forward to the headline when we can say Cincinnati Children's closes its transplant program because we have definitive therapies. We're getting close. So our research goal, when Chunyue, myself, and Aki and Emily put this together, we said what we want to do is to have a time for parents, patients, hearing from all of you, the researchers, physicians, and certainly industry, to connect, to share concepts of disease models. What's the best way to study these diseases? So that we could build new projects in conjunction with all of you, and then highlight the challenges. And we've certainly heard the challenges. Because PFIC is a set of genetic inherited diseases. And trying to understand genetic disease has always been a challenge. So I like to keep things simple. So I have a very simple analogy. If I want to be able to cook my eggs for breakfast, that requires several steps. There has to be a producer of a protein that has to be delivered, and that has to function. Well, PFIC is very similar. There's a gene that has to produce a protein that may have to be chaperoned and delivered so that it can function. And the function in these disorders is to drive bile flow. So our objective with the workshop was to discuss models, how to get to that question, how to investigate every one of those steps. Because for every one of those steps, something can go wrong. There's a defective gene, there's a mutated protein, the chaperone is not directing you in the right direction, or there is some other functional defect. So if we could investigate these genetic diseases, we could then ensure a more precise diagnosis. I've heard from many of you not sure what the disease is. Some of you know exactly what it is. What about then understanding the cause, how that mechanism leads to this liver injury? And therefore, if you understand the cause, you could develop, as Aki told you, develop new novel therapies. My focus, Chunyue's will be on mechanism. My focus is on the diagnosis, because our mantra when we started our Curl Center was every patient deserves a precise diagnosis, and we can do that, or we're getting close to being able to do that. You see a genetic defect. Is this a new disease or is it a variant? Because some of the pathogenic variants in recognized diseases are well known, but others may be what's called VOUS, and that's a very frustrating term. That means it's a variant of unknown significance. And as a patient and parent, that's the last thing you want to hear. You know what its significance is to your child. Why can't the genes tell me better? Once we know that, will these variants have a functional consequence? Are they causative? of the problem. And to do that, you need experimental model systems, and that's why we got so deep into models. And the tools that we heard used during this workshop were multiple: human, animal, zebrafish, using sequencing, you've heard about that, modeling mice or zebrafish, in vitro or in glass studies, using such things as organoids, and then other models. So our speakers, and first was Dr. Rodrigo Florentino from UPMC in Pittsburgh. His model was a human model, and he was able to use a technique called a knockout to create, using stem cells, a very nice model of PFIC1 deficiency. He had cited the problems that we had, and which has impeded scientific discovery. We can't advance until we understand the problem. So what he did was he explored how these pluripotent stem cells, in other words, the very basic cells that make up our body and then are directed to different organs or structures, how can they be used to generate patient-specific liver tissue to model PFIC? And the bottom line, he created a biobank. In other words, he's got a bank of human tissue that then he can ask questions of, he can interrogate, and therefore was able to bioengineer a human programmable liver, a great tool, key, available to all of us. So thank you, Rodrigo, and if you have questions and comments, we'll bring it up in a minute. Dr. Sylvia Villarino from Yale used that same stem cell model to talk about a new type of PFIC, a new mechanism, and that is patients with what's called KIF12 variants. This is a model of kinesin, and that's a chaperone, basically. Tells proteins where to go. And she says this is a new form of a cholangiocyliopathy. In other words, a mechanism that hadn't really been described before. She found that very rare variants in KIF12 cause high GGT cholestasis, just like, say, MDR3. This is a family member of microtubules. Microtubules are basically the highways in which things move across cells, and it's expressed in bile duct cells. So she generated these human pluripotent stem cells, biliary models, with and without mutations, so she could see the difference, what causes the problem, really understanding the mechanism. And she demonstrated that these KIF12 mutant cells have increased GGT activity, like MDR3 deficiency, and organelle mislocation. In other words, the chaperone was leading them down a blind alley, sort of like a traffic jam. So it was impairment in organelle trafficking. So just like a defective gene, if you can't get the protein in the right place, it's not going to work. So clearly new insights into human cholestatic disease, another model. Thank you, Dr. Villarino. Hank Verkade from University of Groningen used the mouse model. And his mouse model was not a knockout, but a knock-in model, to create a human-like mouse. In other words, he had a gene inserted into a specific locus into the genome. So a human-like mouse that he could then study to see how this disease would affect the individual, and more importantly, how he could manipulate that to develop therapies. His model was a knock-in E297G, one of the more common PFIC variants, and it produced an immature BSEP protein. And the model had BSEP disease, basically, elevated liver enzymes, elevated serum bile acids, and inability to excrete bile acids in bile. So this tool allows quantification of BSEP function, transport capacity, and allows studies of the intervention, that humanized mouse. So clearly teaching us about various variants, whether they're missense or, as Aki mentioned, deletions, truncations. And then last part of this section, Chunyue talked about her zebrafish. She's got all these zebrafish that she can use as experimental models to study new PFIC genes and variants. The lamentation that we deal with every day is only a small percentage of our patients with known liver inherited metabolic disease have mutations in known PFIC genes. So using new techniques, whole genome sequencing, whole exome sequencing, has clearly accelerated new candidate genes. But do these genes mean anything? So to validate that they really cause disease, these novel variants, is a challenge and therefore hinders personalized treatment. So what she has done is she's developed a zebrafish system to functionally characterize any mutation. So she can look at the zebrafish model and see what the effect of a mutation has. As an example, she tried to develop a model of a new form of PFIC. It's a deficiency in a novel protein, MRP9, and it causes what looks like any other form of PFIC. This was a paper published in 2021, and she found this was related to ABCC12, a new form of PFIC, and thankfully she did not give it a number. What this did, using the zebrafish, it was started with the cells from a human expressed in a mouse model, and then in her zebrafish, she found that MRP9 deficiency renders bile ducts susceptible to injury, the cholangiocytes. So in the wild type, or normal, you can see the MRP9 protein sort of ices the cake of the cholangiocytes and keeps them healthy. In the absence of that icing, the liver cell, the cholangiocytes will undergo apoptosis, programmed cell death. So a very nice model that, again, taught us that if you keep looking for these new disease models, you'll find it. So the bottom line, Chunyue, keep searching. You may not find Nemo, but perhaps you'll find PFIC type 99 and not call it PFIC type 99. Take over, Chunyue.
So, sorry, Dr. V, I have to admit, we actually call ABCC12 PFIC 5. Then I realized PFIC 5 was already taken.
You should listen to me.
Yeah, you dig our refrigerator, you find tubes that PFIC 5. That's ABCC12 samples. Sorry. First of all, I want to thank everybody here, and then I call this meeting Pick Me Up meeting every year, and I got motivated to do work like Rodrigo said. And actually, when I say goodbye to Dr. Grompe, he said same thing to me. He's more motivated than 2 days ago, so to do his research. So thank you so much. I don't have nice slide like Dr. B has, but it's all words. I want to summarize the 3 talks on new promising therapeutic strategy for liver disease. So the first talk is from Dr. Stacy Herbert from Cincinnati Children's, and she used RNA-targeted medicine. So I remember Dr. B's egg example. So DNA make RNA, RNA make protein. So her strategy is to target RNA so that you can manipulate the protein expression. And this method is rapid. You deliver RNA, you see the effect really fast, and it's reversible because RNA is not in your genome. It gets degraded, it goes away. So she's interested in studying Alagille syndrome, which is caused by mutation in the JAK1 or NOTCH2 genes. And then these patients also have cholestasis. So her group designed the antisense oligo to basically knock down a RNA called POPO-GLU2. So this protein functions to manipulate the Notch protein activity and interaction with its ligand. So she showed that by delivering this antisense oligo to a mouse model of Alagille syndrome, it knocked down the RNA of this target gene and then also rescued the bile duct loss. But from my complicated description, this method really relies on understanding the biology of the disease and then what this mutation is doing to the protein. So this is really rely on the science part. And then the second speaker, Dr. Marcus Grumpe, talked about cell and then gene therapy. The general idea of cell therapy is to deliver healthy hepatocytes into diseased liver, but the challenge is the efficiency. You cannot get enough healthy hepatocytes engrafts in the liver to resume basic liver function. To overcome this, he generates hepatocytes that are resistant to Tylenol, which everybody knows what Tylenol is. So when they put these cells in mouse and give these animals low dose of Tylenol, the engineered hepatocytes have advantage over the donor cells because they are resistant to Tylenol-induced toxicity, and therefore they can engraft and populate in the donor liver. Then when you put wild type gene of interest, PFIC gene, in these cells and put them in the patient liver, then these cells will grow and then hopefully will cure the disease. So he also comment on there's a potential use of this strategy on PFIC type 3, which is MDR3 deficiency. So last talk by Holger Willingren from UCSF. His lab actually screened through over 100 AAV vector. That's the vector used to deliver gene therapy to liver cells. He screened these AAV vectors, and then he found different vectors will target different liver cell types. So some forms of cholestasis, including some PFIC, like the ABCC12 I studied, and then the KIF12 is actually hitting cholangiocytes, not hepatocytes. The problem is most of the AAV vectors hit hepatocytes, not cholangiocytes. So luckily, he found one very specifically targeting cholangiocytes. So this new tool provides potential to deliver gene therapy to the cholangiocyte disease. So that's kind of my summary of what I heard. I think for gene therapy, the technology is there to effectively deliver gene to liver cells, and it's still quickly evolving, but there is room for improvements for the viral vectors, and like how long they stay there, and then the immune response, and then how to increase cell type specificity. And the most important is the safety. Now we have these mouse data, how can we push further to the next stage is really need to demonstrate the safety. So that's all I have here. I hope it's not too much. Okay, any questions for us here?
Or comments? I think you'd agree, Chunyue, that the future is bright and hope for not only better diagnoses, but strategies for us to develop new therapies without question.
Yes. So I will say I see lots of familiar faces. These families, they come every PFIC meeting. So I hope, every time you come here, you get more hopeful and more encouraged. Thank you so much.
Thank you all very much. That was a really fantastic summary. Thank you for doing that. It really does drive it home, all of the phenomenal things that you talked about in your work, and made that very relatable. So thank you. So I'm going to go ahead and just close us out. We skipped our break, and so I feel like I owe that to you. But really key takeaways here. You've heard some really amazing things today. We've had some really amazing engagement. We were a little bit vulnerable together. I think that we've really built a community amongst patients and researchers. So I feel like now when I say the word PFIC community, I think of this room. It's not just my peers as patients and parents, because I really see how invested the group of people who are in this room is in the disease, whether you are a patient or a parent or a doctor or a researcher or none of the above and you're in this room, you really are our community. And so thank you for being here with us. A few opportunities to get involved and stay involved. We highlighted them throughout the conference. These are very specific research opportunities. Obviously, the PFIC network has a lot of family support opportunities as well. But we talked a lot about the patient registry, and I think we all see the potential in the registry, especially if we sign up and we stay engaged. So we do the raffle twice a year. The next raffle we're doing is in May. The registry is available in 4 different languages, if you include English. The way that we engage families in the registry is by email. You'll get your follow-up every 6 months by email. Check your spam folder. A lot of people don't see their email, and so we'll work through that. But mark your calendar. Let's try something new. Or just message us. I can't tell you if you're in the registry because I don't have privileges to see who's there, but I can get you in touch with those to see if you're enrolled. So just please get involved and stay involved. Project Impact. I think you saw the value of not only the discussion in the focus groups and how we can talk to each other about very specific things, about what impact is actually doing and what impact will be doing over the next 1, 3, 5 years, really. So join us in Project Impact. The Treatment Experience app is going to launch May 9th. The beta version's available. If you're interested in testing it out, contact Melissa. We'll get in there and you can take a look before May 9th. But join us in that initiative. We do modules and focus groups for impact. The modules take 10, 15 minutes, and they're actually quite educational. So if you enjoyed the dialogue here, please participate in the modules. They're quite simple. And then we do the focus groups every 3-4 months, I think it is. So our next bi-monthly. Oh, sorry, yes. Yeah, bi-monthly. So the next one will happen in June. Thank you. And then all previous impact modules are available on our website. And when they launch, if you're one of the first 30 participants, you do have a very small financial incentive to join that initiative. So that is the way to stay a part of the conversation ongoing until we make it to next year, where we are going to participate in a partnership for the first ever cholestatic liver disease summit. We're joining in, again, with the Alagille Syndrome Alliance and then with the biliary atresia group. And there's going to be a few other groups, PSC partners and another biliary atresia group, and potentially a few more who are going to come together, and we're going to do a combined event, but we're also going to have breakouts to where we focus individually. And so we're going to do that next year in Aurora, Colorado. It is very near Denver. And so please join us June 26th to 28, 2025. And you'll hear more about that coming up. From our community, which is a global community, this is the patient community. Thank you for being involved. Again, thank you very much to our partners in this event, Cincinnati Children's Hospital. You've been wonderful partners, and thank you for helping us provide this opportunity here. The PFIC network team really is what keeps this bus going. So I know I say you made my work very easily. You made our work very easily because I can't do any of this without the support of my team. And so we have a wonderful staff at the PFIC network, and they really help to keep us going. And I know that you've been engaging with them over time, and you will continue to engage with them. But this is really the team that makes all of this happen. And thank you for joining us. We'll see you next year.